Hi everyone, tonight's video is on linked genes. So I want to look at a cell undergoing the process of meiosis. We're going to start with a cell that is heterozygous for two genes. It has both the dominant and recessive allele for the R gene and the dominant and recessive allele for the Y gene. The R gene is found on the larger of the two chromosomes and the Y gene is found on the smaller of the two chromosomes. Once these chromosomes have undergone replication and have lined up on the metaphase plate at metaphase 1, they can line up in one of two ways. They could either line up so that both of the dominant alleles were on the same side and the recessive alleles were on the opposite same side, so that at the first meiotic division, the two cells that arose, one would have both of the recessive alleles and one would have both of the dominant alleles, ultimately resulting in only two types of gametes. Either two would have both of the recessive alleles and two would have both of the dominant alleles. Alternately here at metaphase 1, the chromosomes, the homologous chromosomes, could line up so that one of the dominant alleles was on one side and one of the recessive alleles was on the other side. In this way, after the first division, each of the cells would have one dominant and one recessive allele, and then when the four gametes were produced, they would each be heterozygote. The chances of them lining up one way or the other are completely random, and we refer to this as independent assortment or independent orientation. Both are correct. And that has to do, or that means that tetrads line up at metaphase 1 randomly. They can flip-flop from one side to the other, and it doesn't mean that always the dominant alleles are always on one side. It's random as to which side the tetrads line up. This results in random combinations of alleles found in each haploid gamete because the alleles segregate independently. The alleles that are on separate chromosomes can line up differently and they can segregate into different gametes. But what do we see in the real world? What's going on here? It looks to me like blonde hair and blue eyes seem to go together. Most of the people I know with blonde hair also have blue eyes. Dark hair and dark eyes seem to go together. Again, most of the people I know with dark hair also have dark eyes. When we have two characteristics that tend to sort together, we refer to these as linked genes. So how do linked genes work? Well, let's look at the number of different genes on a chromosome. This is chromosome 10, and this is just a short-lived list of the different genes that you would find on this chromosome. Every chromosome has hundreds of different genes, with the exception of the Y chromosome. Genes are in specific locations on the chromosomes, and those locations are called loci. So genes on the same chromosome should stay together during meiosis. All of the genes on the same chromosome, in fact, should be linked, but they're not. Why? Let's look at the process of crossing over. During prophase 1, the tetrads or homologous chromosomes pair together, and you get what's called a chiasmata, or a crossing over, where the chromosomes actually can trade parts of the chromosomes in between homologous pairs. And this means by the time anaphase 1 occurs, this blue chromosome now has a piece of the red homologous chromosome, and conversely, the red chromosome has a piece of the blue chromosome. Crossing over always occurs. It occurs in prophase 1, and it's not a rare event, it's a 100% event. However, where the crossing over occurs on the chromosome is a random and statistical event. So why are blonde hair and blue eyes linked if there's always crossing over events in between on the chromosome? Doesn't that mean that all of the genes, even on the same chromosome, could be separated from each other in meiosis? Well, remember, crossing over is a random event. So if we look at a pair of homologous chromosomes, here's my tetrad, and let's look at two different genes on these chromosomes. We have the G gene and the B gene. This chromosome has the dominant G, and this chromosome has the recessive G, and conversely for the B gene, we have the dominant and the recessive. The likelihood that a crossing over event could occur somewhere in between this B and G is very, very high because there's such a large amount of real estate in between these two genes. So the chance that you're going to have at least one crossing over event in all of this space is very, very high. However, if we looked at a different situation where the G and B genes were very close to each other on the same chromosome, in order to be separated by crossing over, you would have to have a crossing over event occur right here, right in between those two genes. And that's very unlikely. It's much more likely that you're going to get crossing over events somewhere in this large chromosome, but to have to get it right in between these two genes is very unlikely. So if they're very close together on a chromosome, the likelihood of a crossing over event is very small. If they're very far apart, the likelihood of a crossing over event between them is very high. So linked genes are genes that tend to sort together at anaphase 1, and by definition, they have to be close together on the same chromosome. Just being on the same chromosome isn't enough, because if they're far apart on the same chromosome, the likelihood of a crossing over event between them is very high. They have to be close together on the same chromosome. We could look at something called a linkage map to show the statistical frequency of linkage between pairs of genes. The closer two alleles are together on a chromosome, the tighter the linkage. So let's look at this gene. This happens to be a gene for a fruit fly, and so one of the genes is called Aristillus antenna, and I don't know what that is. But if we look at the gene for curly wings, the Aristillus antenna and curly wings only separate from each other, are only unlinked 6.1% of the time. That means 94% of the time, these two genes are linked. They tend to sort together. However, alleles that are far apart on a chromosome don't behave as if they're linked at all. So let's look at how often Aristillus antenna and brown eyes sort together. And you can see that 100% of the time, they do not sort together. They are not linked. They go separately 100% of the time. They are so far apart, they may as well be on different chromosomes because they simply do not sort together. There's always going to be a crossing over event somewhere in between each end of this chromosome. So does that mean that alleles that are right next to each other never unlink? No, it is a statistically random event, so even genes that are very close together could unlink, but it would be very rare, and you would see it very rarely in a population. So from tonight's video, you should know, what is independent orientation or independent assortment? What is the definition of linked genes? Why aren't all genes on the same chromosome linked? What's a linkage map? And experimentally, how would you identify linked genes? So that's all for tonight.